In this second screencast, we're going to be focusing on DNA replication with a little review of what the DNA molecule is all about. So this is review. The DNA structure is really important to understand to grasp all of the actions that it plays a role in through genetics. So remember that it's made up of subunits. It's a long polymer. It's made up of subunits called nucleotides, and each nucleotide contains these three things: the phosphate, the sugar, which is specifically deoxyribose, where the name comes from, and then a nitrogenous base. There are indeed 4 different types of nitrogenous bases, and in those nitrogenous bases, we can just remember these letters. They work for us: A and T, which are complementary, and C and G: adenine, thymine, cytosine, and guanine. This characteristic of complementary base pairing is really important to the structure and, again, the functioning of the DNA molecule. Remember that A and T always go together. They will always form the opposite rungs of this quote-unquote ladder in a DNA molecule. A and T, C and G always go together. So if you are given one of the strands of a DNA molecule, you should be able to give me what is called the complementary strand, just by knowing the complementary base pairing rules. As a reminder, this relationship between DNA and chromosomes is really, really important. The genetic material is always found in the nucleus of a cell, and it comes in a couple different structural forms. The chromosome is basically just wound up DNA. It's like a roll of tape. It's like a ball of string, where the DNA is like the individual strand of tape or the individual string. So just to dive in and touch upon human chromosomes, human genetics, and the role that genes and DNA play in the traits that you have in the future here, just want to mention what DNA really means in your cells for now. This is a picture of human chromosomes, and we have 46 total chromosomes, but usually I refer to them as 23 pairs. The reason they occur in pairs is that for every pair, you had one from mom and one from dad. If you see this, there's 23 different pairs of chromosomes. Of each pair, one came from mom, one came from dad. So literally, physically, what makes DNA the transferable material is that you will have 23 chromosomes from mom in an egg cell and 23 chromosomes from dad in a sperm cell. So remember, we took DNA out of strawberries, a DNA extraction out of strawberries, and these are just some statistics. They're not important to know, other than we know that DNA is a long polymer, a very long, slender molecule, that when clumped up, you can actually see it with your eyes. But when it is all stretched out into a thin line, it's quite small. Just a couple more numbers to wow you with how much DNA is actually there in your cells. So the process of DNA replication is really simple. Replication means to replicate, to make a copy of itself. And we're going to keep it actually quite simple. And at its basics, it is simple. It's DNA just making a copy of itself. If you think about it, there's a couple different reasons for why DNA actually has to replicate or make copies of itself. A couple of the perhaps obvious reasons are that when you make new cells, when a body makes new cells, and since all cells have a total copy of your DNA, all of your cells have all 23 pairs of chromosomes in the nucleus of them. Every time you make new cells, they have to be genetically identical to the original. So a couple reasons for that: when you were a couple months old, like this little baby, you definitely had less cells than the 80 to 100 trillion cells that you have today. So to grow is to grow more cells. Or if you fall off your bike like this little wimp did, this little kid did, and just kidding, and get a cut, get a damaged tissue, get a broken bone, all repair of damaged tissue isn't just some magic glue. You actually produce more cells to heal the damaged tissue. And for our purposes, it really is this simple. It's a 3-step process that is pretty simple. DNA replication, if you look at a DNA molecule, remember it's a twisted double helix shape. At the right timing, which is all, again, run by genes and DNA in the first place, but the DNA molecule is going to unzip at what's called a replication fork, and an enzyme called DNA helicase is the enzyme that actually does the untwisting. Remember, enzymes are things that get reactions done in the body. So helicase unzips it, and the new nucleotides, the simple third step, is that new nucleotides are added, of course, complementary to the old strand, or the original strand, to build 2 new and identical, they're called daughter strands of DNA. The molecule, again, is an enzyme. It's called DNA polymerase, specifically DNA polymerase 3, but it is the enzyme that actually gets the job done. It actually is the carpenter swinging the hammer, pounding nails to get this thing done. Enzymes get stuff done. And as you see, there's a lot of more complicated chemistry and action going on here with the directions of the growth that happen opposite. There's 2 replication forks, but really these are the 3 steps to know. This is just showing you, this slide is showing you that an old strand unzips and separates, and both sides act as a template for the new strands. There's nothing new, there's nothing random about which bases are going to go where, because remember, A always will go with T and C will always go with G. So ideally, barring any mistakes that might happen, you are going to end up with 2 DNA strands, or in other words, 2 DNA ladders that are identical to each other and identical to the original. Now the color makes it look like they aren't identical, but genetically, the order of the bases should be identical. Same process, take a minute to look at the labels and to see what's happening and to remind yourself and review in your mind the 3 simple steps that we've talked about for replication. Something else to add to this is that these free-floating nucleotides is what they're called. These free-floating nucleotides are already available in the cell, and they come from recycled DNA material. They come from recycled nucleic acids that are in the cell. They come from raw materials from the food that you eat, and you digest it into the small usable building blocks that we've talked about before. So those are referred to as free-floating nucleotides. They're already available in the cell for the polymerase to add to the growing daughter strands. It's important here to introduce what's called a mutation. There's a lot of different types of mutations, but mutations can occur during DNA replication. So we just got done talking about the process of DNA replication, where you're going to end up with 2 ideally identical strands when the whole process has ended. However, this is life science, this is biology. There can be, and often are, mistakes in the DNA sequence during replication. And you have a couple different views here. I mean, there's a lot of different causes of them, but sometimes there's just, for some reason, a mistake in building the new strand. And one of the most common mutants or mutagens is UV light, and it can cause skin cancer. It can actually alter the DNA in skin cells, and therefore a lot of times what that causes is uncontrollable and unchecked growth in the skin cells. Well, uncontrollable growth, unchecked growth is the definition of cancer. There's different types, and this little screen down here gives us an example of, or an analogy of, types of mutations. And the whole words and letters and sentences is a great analogy for us to use. And you see a couple different types of actual mutations in DNA in cells, and you see what happens to words and sentences. So if the sentence is, "As the man saw the dog hit the can," you can then see what happens if you alter letters or rearrange words or get rid of words completely or bring brand new words into it. You simply change the overall outcome or message of the sentence. A simpler example, if you take the simple word cat, if you rearrange those letters in any other way, it no longer means cat. I use the exact same 3 letters and rearrange them, I get a completely different word with a completely different meaning. Now, the big picture story of that is that if DNA is the original code, original recipe, original blueprint for certain traits, if you mess with the code or the recipe or the blueprint, the finished product has the potential to be changed, and it can be changed quite radically. So in review, we took some time to go over the DNA. The DNA structure is important to just have that down. Be an expert in DNA structure. DNA replication is the simple process of making new DNA. You simply make copies. You end up with 2 strands where you used to have 1. But make no mistake, there is nothing random about DNA replication. There might be mistakes, but there's nothing random about the complementary base pairing rules in DNA and DNA replication.